Welcome to the development of B cells module. In this talk, we're going to have an overview of B cell development. We will begin with learning about lymphopoiesis and describe the origins of B cells. We will then look at the different stages that are involved in the development of B cells. Lymphopoiesis is the production of new lymphocytes, and lymphopoiesis normally occurs in the central lymphoid tissues, which are also called as the primary lymphoid tissues. These include the bone marrow and the thymus. Now, in the bone marrow, when we look at the types of lymphocytes that are made, B lymphocytes go through the process of lymphopoiesis to a much later stage than the T lymphocytes. The T lymphocyte progenitors are made in the bone marrow, however, they migrate to the thymus and undergo maturation there. It should be remembered that all lymphocytes arise from hematopoietic stem cells that are present in the bone marrow. So regardless of where the maturation is completed, the origins for all lymphocytes are the hematopoietic stem cells that are found in the bone marrow. What should also be noted is that the bone marrow has an environment which has non-lymphoid connective tissue, and these are made up of cells called stromal cells. Stromal cells play a very important role in lymphopoiesis because they provide the appropriate signals that are required for the development of the lymphocytes during the process of lymphopoiesis. B lymphocyte development can be divided into three parts. The first is the stages of development that occur in the bone marrow, followed by the screening mechanisms that enforce tolerance, and finally the development of the immature B cells in the spleen. All lymphocytes, as I told you before, arise from hematopoietic stem cells, and that is what is shown in the black box in the figure to the right. These hematopoietic stem cells are able to differentiate into another type of a progenitor cell called as the multipotent progenitor cells, or MPP. The MPP cells can undergo further differentiation and form progenitor cells that can be either of lymphoid or myeloid origin. The figure that's shown in the black box shows the common progenitor cell that will give rise to myeloid cells, and hence it is also called as the common myeloid progenitor. The common lymphoid progenitor is another progenitor cell that can be formed from the multipotent progenitor cells, and the common lymphoid progenitor is the one that will form NK cells, B cells, and T cells. So all our lymphocytes, including the B lymphocytes, arise from the common lymphoid progenitor. Let us have an overview of B cell development. Once the common lymphoid progenitor is formed, the common lymphoid progenitor can commit to B cell development by forming an early pro-B cell. In the early pro-B cell, we can see the beginning of somatic recombination in the heavy chain. The early pro-B cell differentiates to the late pro-B cell. In the late pro-B cell, we see completion of the heavy chain recombination. The late pro-B cell will differentiate to the pre-B cell. The pre-B cell can come in two parts, which are the large pre-B cells, which will then differentiate into the small pre-B cells. The small pre-B cells will then differentiate to form immature B cells, which are able to express the membrane-bound immunoglobulin M, or IgM. The immature B cells will go through a screening process, and those that have been able to get through the screening process and survive the process will then leave the bone marrow and go into the blood. From the blood, they will eventually go to the spleen, where the immature B cells will then go through a transition 1 and transition 2 state of differentiation to ultimately form mature B cells. With this, we come to the end of our talk, where we learned about lymphopoiesis and the origins of B cells. We also had an overview of the different stages that are involved in B cell development.